Good morning, everyone. Welcome to the MedTech Lounge, where we talk about the latest trends, advertisement, and important information in the world of molecular biology. I am Kyla Torres, your host for this segment. So for today's segment, we invited Miss Camille Santos and Miss Angela Nalog to help us discuss and know their personal stand on gene cloning. At this point, everyone probably heard of cloning. A clone is a genetically exact copy. It can be a clone of a gene, a cell, or an organism, even a human. However, whereas cloning of humans has many ethical issues associated with it and is illegal throughout most of the world, the cloning of genes has been ongoing for well over 30 or 40 years, with the cloning of animals occurring more recently. What I would like to know is your stand on this matter. Because personally, I support gene cloning since it benefits both people and animals in several ways. It can aid in boosting food production and assisting child-wanting couples. Its significance stem from the capability to produce DNA fragments containing a specific gene which has enhanced our comprehension of the structure, function, expression, and control of that gene. What about you, Angela?
Before that, I would like to present some pros and cons of gene cloning. When Dolly, the first clone sheet, came in the news, cloning interested the masses. Not only researchers, but even common people became interested in knowing about how cloning is done. Consequently, scientists realized that it can be used to bring about desired changes in the genetic makeup of individuals, thereby introducing positive traits and removing negative traits. Cloning could be used to develop human organs, potentially making human life safer. But like every coin, cloning has its flip side too. Despite its advantages, it's unclear whether humans will bring all the potential uses of cloning into reality, as there is always the risk of abuse, development of new diseases, and decreased genetic diversity. And so, I think that gene cloning has played an important role in fundamental biological research. Its value lies in the preparation of DNA fragments containing a specific gene, which has resulted in advances in understanding the structure, function, expression, and control of that gene's activity. It also has the potential to improve veterinary and human medicine, with the possibility of non-reproductive human cloning strategies providing future therapies for severe and incurable diseases.
But what about its effect on society? Does gene cloning potentially change the course of our future?
Hi, good morning to all of you. I'm Camille Santos. DNA molecules built through cloning techniques are used for many purposes in molecular biology. In biopharmaceuticals, recombinant proteins are used to treat strokes, prevent blood clots, produce insulin, and synthesize human growth hormone for individuals that need it. Also, it is utilized in gene therapy for the delivery of plasmids by gene cloning to cystic fibrotic patients, which lessens the deterioration of the lungs. Even proteins, vaccines, and antibiotics are all produced by the process of cloning. Moreover, in agriculture and livestock, cloning is employed to create pest-resistant plants and transgenic animals. It is a way to duplicate desirable features like rapid development and abundant milk production without the genetic lottery. Plus, much of the criticism of this technology is actually a criticism of the abuse of modern technology and the wrong business practices of huge corporations. But on a larger scale, gene cloning and genetically modified organisms can be part of the solution to make agriculture more sustainable and understand the biological processes of animals using tests.
Certainly, I agree with you, Miss Santos. With all of that said, we would like to thank Miss Angela Nalog and Miss Camille Santos for providing us answers about gene cloning. And for the viewers, thank you so much for tuning in on the MedTech Lounge every Saturday at 7 a.m.